Hello, everyone, and welcome to today's webinar, Ask the Scientist, Soil Health, Fertilizer Reductions, and Farm Profitability, which is presented by Holganix. I'm Robin Sitberg of Meister Media Worldwide, publisher of CropLife magazine, and I'm moderating the webinar today. The webinar has been approved for a half CEU credit in soil and water management from CCA. To receive credit, you must watch the entire webinar. There will be a QR code at the end of the presentation that you can use to submit your credits. We will have time, as always, for some questions at the end of the webinar. So if at any time during the presentation you'd like to ask a question, go ahead and type it in your question pane, and you can do this at any time during the webinar, and we'll answer questions at the end. Before we begin, I'd like to introduce today's speaker. Dr. David Stark is a molecular biologist and biochemist who holds 9 patents and has 26 peer-reviewed scientific articles. His past career includes leadership positions in Monsanto's technology and business units. David became the president of Holganix Agriculture in 2015 to lead the company's efforts in the agriculture space, including commercial development, R&D, and overseeing various studies. So now I'm just going to go ahead and turn it over to Dr. Stark.
Okay. Thanks, Robin. Thanks, everyone, for joining. As she said, I'm a Ph.D. molecular biologist. I had my doctorate from Washington University in St. Louis, and my undergraduate degree is from The Ohio State University, where I know other people on this call, like our moderator, also went. So that's why I bring that up. So, you know, just to kind of set the stage a little bit, I spent my first career at Monsanto and started as a scientist, went into management and such. When I retired, so you don't see my face, which is on purpose because my hair is no longer dark or as thick as it used to be. But when I retired, I started consulting, and a friend of mine, actually from church, asked me if I would mentor this little company called Holganix. They were actually an existing turf company, but they wanted to get into agriculture. They were stumbling across agriculture. And I said, sure, until I found out what they did. When he told me they made this funky brown bug juice, I thought, no. So my experience up until meeting Holganix was that microbial products were just not that great. There wasn't enough science around it to really convince me. But I did it anyhow because my friend asked. And now look what happened. You know, 9 years later, I'm still with the company, and I've done a total 180 because I took the time to really understand the value of the soil biology. And it's incredible how much we didn't know that we're learning now. We still have a long way to go as an industry. So part of what I want to do, and I don't have the slide in the deck, but there's an investment group that put together a landscape of 400 different companies selling biologicals and microbials. I know how confusing the landscape is. It's okay to be highly cynical because there's reasons to be cynical, but at the same time, I want you to know the foundation now, the science behind soil health is really good and it's legitimate. So let me just start with this slide where, you know, how many times through the years have you guys been told and have I heard, you want to make your soil better? Reduce your tilling or go to no-till, start planting cover crops, you know, spread manures and other organic material. And through time, you know, trust me, your soil will get better. And I don't know how many guys have tried that, and it's been painful. Sometimes guys find it works great year one, but often I hear it takes 3 to 5 years. It costs money. A lot of mistakes are made. And it's because we're doing it backwards. Because what we have found that if you add life to your soil first, that actually enables you to go no-till. That enables you to have cover crops. That enables the soil to improve, and it doesn't have to cost money year one. So these are the things we're going to start talking about in this presentation, everything behind it. So when I talk about adding life to the soil, and by the way, Robin, let me know if my slides aren't advancing, but I'm on the slide now that has the microorganisms. If we look at the left-hand column, you see different types: bacteria, actinomycetes, fungi, protozoa, nematodes, etc. Then if we look at the relative number per gram of soil, that middle column, and by the way, a gram of soil is about the size of an M&M, a chocolate M&M, not one that has a nut in it, so it's not very much. And so you can see bacteria, there can be over 1 billion in a tiny bit of soil. And as the microbes get larger, slower to grow, you can see that there's fewer. They're all there for a reason. But now let's look at the right-hand column. This is the biomass of these in the top 6 inches of your soil per acre. So bacteria, which again are the smallest, the fastest to grow, the most abundant, there can be over 2 tons of bacteria and actinomycetes in good soil, or a few 100 pounds in poor soil. But look at the fungi, almost 7 tons in good soil, in bad soil, 9 pounds. There's a huge discrepancy. What's going on? Well, let's take a look. So there are things that happen that change the microbes in our soil. And by the way, they're always changing throughout the growing season. That's normal. That's the way God designed it. But there are things we do that change them and things we have no control over. So for example, and we're going to talk about this later in a little bit more detail, when we till soil, we really change the microbiome. So tillage changes it. Some of our practices, so obviously when we fumigate, the goal is to basically sterilize like the top foot of the soil. That's not good. Overuse of fungicides, really overuse of fertilizer. And I don't bash fertilizer. Fertilizer, frankly, is a blessing. But when a plant senses excess nitrogen and or phosphorus, it stops feeding microbes the way that we want it to. So we need to back off. But there are things that happen we have no control over. Winter. It hasn't been that harsh of a winter, but we did have a period in the north where it was very, very cold for about 10 days. Microbes are full of water. When water freezes, it expands. It kills microbes, not evenly. So coming out of winter, the soil is almost always out of balance. Plus flooding. What we want in the soil needs oxygen. 48 hours underwater or water saturated, the oxygen's being depleted. So now the microbes that are beneficial are dying off, but the ones that don't need oxygen, which tend to be the pathogens, can now grow and take over. And one easy way to tell is when the water drains, you can smell it. It's like the bottom of a swamp. That tells you that ground went anaerobic and it's way out of whack. Just to make it more complex, the microbes in your soil depend on what you grew last year, what you're growing this year. In fact, we found 6 to 10% of the microbes in any given root zone not only change between corn, soy, wheat, berry bushes, trees, roses, whatever. 6 to 10% vary from corn hybrid to corn hybrid, from soybean variety to soybean variety. It's because microbes are being farmed by the plants, and we're going to really hit that theme. So my point is, earlier about no-till and such, there are a lot of things we do. We can't always assume that when we're doing things right, that we still have all the biology in the soil. So this is an example of a microbiometer test, and this is something maybe you've heard of. It measures fungal to bacteria ratio, and it looks at microbial biomass carbon. Basically, what's the total biomass of the microbes you have? So if we look at the one on the right, 70 micrograms C per gram of soil, that's really, really low. This is a field that was 15-year no-till. By the way, I'm not saying this is your farm if you're no-till. I'm just saying this is an example of something that, even though the guy thought he was doing something good, his ground is very fungi or bacteria dominant, almost no fungi. He does use anhydrous. And anhydrous, kills everything it touches, but I'm not sure that's the reason why the life is so low compared to a farm a couple miles down the road there on the left, where we have, a huge amount of microbial activity, very high fungi. So too many times I give these webinars and people ask me, so what does your company do? I don't like doing commercials. You're getting continuing education credits. But what we do is we make a product that has all those microbes you saw in that chart: bacteria, actinomycetes, fungi, protozoa, amoeba. They're alive. They grow in a batch. We DNA fingerprint for consistency. We have to refrigerate, which is a key that you're getting living microbes. Frankly, it's the only way we stabilize them and keep them from killing each other by the time you put it down. It's not ice cream. You've got 6 to 8 weeks to use it if it comes in a tote, which we put in an insulated wrap. It has nothing else you need to add. There's sugar in there in the form of molasses, micronutrients, amino acids, kelp extract. We buy and add mycorrhizal fungi, a little humic and fulvic acid. So basically, it's a product that in one shot, easy to use, mix it with anything you put down except fungicide, obviously, that adds life back to the soil. So you'll see a lot of data based on this because it's the best data I've got. But I want to get back to really the science here. So on this slide, there's a couple things I want you to notice. See all the really small print. So for example, fungal bacterial diversity and microbiome complexity predict ecosystem functioning. Yeah, that's English in the upper left corner. But underneath it is the scientific reference of Nature Communications and the first author. What I'm going to show you here isn't my science. It isn't my company's science. I go because of my training to the peer-reviewed scientific literature to show you what universities around the world are doing. And so that's really the best place to get the science. One of the downsides or upsides is if you want, you can access this at your library and online. If you are an insomniac and have trouble sleeping, this will help because it's usually pretty dry. But in any event, all these things and more is what the biology in the soil does. So now I'm going to start with one of the big ones, nutrient cycling. So if you've been told that less than half the fertilizer you pay money for and put on the ground actually goes to the crop in the crop year you put it down, that's correct. Nutrient use efficiency is terrible. These are general numbers. Your farm may be better, depends on your soil type and your practices. But generally speaking, these nitrogen numbers are generous. Phosphorus quickly binds with things in the soil, especially calcium, potassium, same way. So if we look at 2021, 36% of the cost to grow corn in the U.S. was fertilizer. It's a $20 billion market. This means more than $10 billion of our money buying fertilizer isn't doing its job. So how can we do better? So this slide has a lot of information on it. This is a summary of 230 peer-reviewed published studies looking at how nitrogen is used in corn, rice, and small grains. The bottom line is there in green at the bottom. Biologically healthy soils produce greater corn yields per unit of fertilizer input. Why is that? So let's look at the box that's surrounded in black. This is what happens when we do not have a biologically active soil. And this is how I think most of us generally think of fertilizer. We put it on the ground, the crop uptakes some of it, and the rest of it sits there. It's lost, it gets tied up, it's volatilizes away, it washes away with surface water or erodes away. But when we have biologically active soil, it's the situation on the right. Instead of either the nitrogen being taken up or it sits there, what happens is it's grabbed by microbes in the soil and they eat it. And they stabilize it and they cycle it back to the crop. Much less is lost. So the nutrient use efficiency is increased. And that's what that SON turnover is. Soil organic nitrogen turnover. Again, another fancy scientific phrase. Let's take a closer look at what's really going on there here in this slide. So here I like pictures. And this is the way that I say that God designed soil to work. And, just as an aside, but something that's very important to me because I should have known better. If you don't know what molecular biologists do, we are experts at DNA and biochemistry and proteins and such, really life at the molecular level. Evolution is something we are all taught and we have a lot of expertise in, me included. So 40 years ago, I would have said, look at, how natural selection and random chance in nature built such an intricate dance in your soil. And we're going to see this throughout the presentation. And I was wrong, because instead of just trying to get an A in a class and, memorizing and spitting back what I was taught, afterwards, I actually spent time looking at the actual science and the actual data. And mathematicians have a term that's called absurdly impossible. And if I look at the mathematical odds and everything that has to happen through random processes, which, by the way, do not write or create new information. They rearrange it, they change it, they delete it, but we don't know how DNA is actually written. And you have 7 feet of DNA in every cell in your body, and hardly any of it, and I think none, is junk. It's an incredibly intrinsic dance. And again, the mathematical odds of a single small protein coming together randomly is a number with 1 out of a number with 191 zeros, if it's a 150 amino acid protein. So 191 zeros. The total number of atoms in the known universe is a number with 80 zeros. So I must have been a person of tremendous faith 40 years ago to believe a random process could build a protein, because it can't. It's absurdly impossible. We have no clue. The best explanation is one that I had known at the time. I just didn't fully believe in it. And it's the one given by God in Genesis chapter 1. So when Jesus talks about creation, as a scientist, I'm telling you, the data has led me that he actually knew what he was talking about. So look at this intricate dance here as an example. We start with fertilizer, lower left-hand corner. It's usually ammonia or nitrate. The plant can take some of it up, but remember the bacteria, they're kind of in the green as a representative picture. Actinomycetes in that kind of red-orange. That's a close-up. They really aren't that different in size. They're the most abundant. They eat it first. They're always hungry. And it's a good thing they eat it, because where do they live? Within half an inch of a living root. So they immobilize it in the root zone. They take ammonia and nitrate and convert it into protein. That's soil organic nitrogen. Now, it's a good thing because they immobilize it. It's bad because of this thing called carbon to nitrogen ratio. In these guys, on average, for every nitrogen, there's 5 carbons. So they have a very high nitrogen requirement. They tie it up. We don't want to put fertilizer nitrogen on the ground to feed bacteria. We want to feed our crop. So how do we get it to the crop? Well, go over to fungi, all that white feathery material you see in that piece of dirt in the hand. Fungi eat bacteria. They're also full of organic carbon. All that white feathery stuff is carbon rich. So they actually put carbon into your ground more than any other microbe. Their carbon to nitrogen ratio is 3 times lower, 1 nitrogen for 15 carbons. Now go to the bottom middle, the protozoa and amoeba. One of these can eat 10,000 bacteria a day. It's why when you look at the chart, if there's 1 billion bacteria, we don't want 1 billion protists. They can eat 10,000 a day, so the numbers should be less. But too many times when we look at ground, and we did thousands of data points last year in soil, we find zero. These things are absent. Fungi are low, produce are zero. So what happens? The soil engine's broken. The bacteria eat the nitrogen, so you have to keep applying more and more and more, trying to get whatever is there to be taken up by the plant. But when we have the produce, their carbon to nitrogen ratio is 1 to 30. They're like you and me. So they're eating a very protein or nitrogen-rich diet in bacteria, and that amount of nitrogen is toxic. They have to spit it out. They spit it out as ammonia and nitrate. Where? Well, where does the food live? Within half an inch of your root. What are the two forms of nitrogen the plant root takes up the most readily? Ammonia and nitrate. The cycle's complete. The engine's working. This isn't all. I told you God has an intricate dance. This is amazing. Remember, when you're out in your field, there is a farm underneath your feet. Plants are secreting food sources and chemical signals to attract many microbes and repel others. This is one reason why. This is work done out of Rutgers University. And by the way, I deleted about half of the graphics from this slide to make it simple. And it's still pretty complex, I know. So in the ground, the plant roots and all the different microbe types are always putting out biochemical signals. When they sense that there's enough different kinds of signals, meaning that there's a diversity of life, so not just bacteria, not just a few strains of bacteria, but we have bacteria and fungi in different species and such, things change, and it triggers something called quorum sensing. And one of the things that happens is the root tip starts absorbing microbes. Plants are farming microbes because they will eat them through the root tip. Now, when they enter the root tip, the outer coating called the cell wall is stripped off, and that cell wall is rich in nitrogen because it's protein rich. Now, many of the microbes without the cell wall just die. They explode. All the nutrients your crop needs, NPK, S, the microbes, or micronutrients, the microbes also need. So when they die during this rhizophagy cycle, now your crop is not just getting nitrogen, it's getting everything. However, microbes live. They travel up the root to the bud. You see all those red arrows? That's the bud where the root hair will form. They secrete a hormone that induces elongation of the root hair. They travel along the root hair. They're spit back out into the soil where they remine the soil for nutrients, reform their cell wall, and it starts again. Plants farm microbes because microbes mine the soil. Now, even to blow this a little bit more in mind-boggling, some microbes survive even further and go to the above-ground parts of your plant because some plants, or maybe all, have hairs on their stems, hairs on the leaves. They induce those. They're spit out. They colonize the above-ground part to help protect your plant, your crop, from pests and pathogens. So the soil biology does all that. How do we know? So again, I'm a scientist, did my doctorate in Missouri, which is the show me state. I like proof, and I like proof like this because it's free. All you need is a shovel and a little bit of your time. So I can tell you for years, guys would call me up and say, Dave, I used your product. The nodulation on my beans is so much better. And I always thought, that's great, but we're just dumb lucky because I don't know why. Now I do. It's because our product is a quorum, because it has over 2,000 species of bacteria, fungi, etc., actinomycetes. It immediately triggers quorum sensing in the rhizobia and other bacteria, because it's not just rhizobia that form your nodules. Now we're absorbed by the root and we get better nodulation. Here's another, in corn, again, all we need is a shovel to look at what's going on. So this is a picture that came out of Iowa. And the grower that sent this to us said, look, where I used your product, the corn is darker green, it's bigger, it has better brace roots and a lot more root hairs. And that's fantastic. But when I saw this picture, so the roots in the upper right-hand corner that say nitrogen only, that's just starter. And it's hard to really zoom in because of the picture quality here in the presentation. There's almost no root hairs, and there's very little dirt stuck to the roots. Where he added, instead of a pop-up fertilizer, a strain of free-living nitrogen-fixing bacteria. And you know there's some out there, and I'm not going to say the company, that's not my point. But 1 or 2 bacteria is not a quorum. So again, bare roots with no root hairs. But where they used our product that has a quorum, now the root hairs are exploding because remember, now they're being absorbed. They're inducing root hair formation. Root hairs and the microbes, when quorum sensing happens, start making a glue. And they glue the soil to the roots. That's called the rhizosheath. And that's what we want to see on your monocots, on your corn and grains. It's because that rhizosheath is where the action is. The nutrients are exchanged. The plant is protected from desiccation. It's where nitrogen fixation would occur. We want to see that rhizosheath. That's the sign of a healthy soil. So just looking at this picture, I can see from the nitrogen only and the nitrogen fixers, this is not a very lively soil. It's lacking biological diversity. It needs to have life because we're not seeing root hairs and rhizosheath. And it was free. All we needed was a shovel. The same thing happens with P and K. So microbes unlock the P and K and feed your plant. One of the things that we want people to do when you're going to use a diverse biological, remember, too much N, too much P, the plants aren't going to farm the microbes as well. So we want it to back off fertilizer. So again, we do soil tests. And here's something, July 11th, where corn was probably planted early to mid-April in northern Illinois. And we're just showing the customer the nutrients are increasing. It's okay. Back off the fertilizer. Let the microbes do their job. Now, as kind of an aside, but it affects nutrient use efficiency, a lot of products out there will give you better roots. And this is because a lot of different microbes secrete hormones that induce rooting. Remember, the root zone is their home. This is important for a number of reasons. In a typical root mass, only 1% of soil is explored. Only 1%. A lot of nutrients need to be close to the root to be taken up. More roots, more root hairs, more soil explored, better nutrient use efficiency. But beyond that, when those roots die, remember, they're making air and water channels, and we need air and water, not only for your crop, but for the microbes. They also eventually become the stable organic matter. So if you want to improve the organic matter of your soil, roots are far more important than things we leave on top of the ground. There's another source of nutrients, which is all that crop residue that hopefully many of you are leaving on the ground. So, just look at corn. For every 40 bushels of corn, there's a ton of residue, a ton of stover on the field. For every ton, there's 17 pounds of nitrogen, about 4 pounds of phosphorus, 34 pounds of potassium, and 3 pounds of sulfur. These are nutrients you've already paid money for. Not a single speck becomes available unless a microbe breaks it down to a basic chemical form the plant root can take up. So how do we do that? Well, first of all, let's go back to carbon and nitrogen ratio. So remember, bacteria, 1 nitrogen, 5 carbons. Your corn stover is 1 nitrogen for 55 to 60 carbons. Wheat and small grains is 1 to 80. It's a very nitrogen-poor food sources for microbes, especially bacteria. Second, though, bacteria generally don't have the right biochemistry to degrade what makes residue so strong and hard to break down. Lignin, cellulose and lignin, but lignin in particular. What does break down lignin more efficiently are fungi. And there are certain kind of class of fungi, they're called saprophytes, but regardless, it's fungi. So you can see that yellow corn, you can put nitrogen down. If you have a bacteria-dominant soil and there's residue out there, remember, they tie nitrogen up, and especially to try to eat a low nitrogen food source that's just carbon, they got to steal nitrogen from somewhere. So we need the fungi. This is a picture of a product that we make that is very heavy in the right kind of fungi that can degrade residue. And there on the left, you can see after a couple weeks, what you want is that black, you can see white fuzzy stuff. If you go in, even some blue fuzzy stuff, it's growing mostly from the inside out. This particular farmer wanted to actually measure nutrients being released from the degradation of his residue. And you can see on the right-hand side where he didn't use the product, and you can see there's no degradation of that residue. There's no fungi. He's deficient in a number of nutrients, especially PK and magnesium. And on the left-hand side, we can see the nutrients starting to creep up because now they're being released. Now, in the southern part of the country, we're following a winter wheat crop with double crop beans. I know wheat straw can be really hard to degrade. This is a great way to return those nutrients to support the growth of the double crop beans. Now, this is a fall application to control residue. This is in Iowa. This is the same field. Part of it was treated with our product there on the right, part was not. This is after 3 weeks. Everything was vertically tilled. I mean, here you could actually drive by and see the difference. But what's important is the conditions were excellent for degradation. Every microbial product, especially fungi, needs a little temperature and some moisture. So if it's bone dry, not even dew in the residue mat, or if the ground's frozen, nobody's microbes, whether the ones you've got out in the field or anything you buy, including ours, is going to do much. We need a little temperature and moisture. Here the conditions were really good. As a matter of fact, they were almost too good in this case. So this is the field in the spring. He didn't plow. Obviously, he could plant right into it, but he felt that he had too little residue cover. So in the fall, when he uses this again, he won't vertically till. It's really kind of a call. What are your conditions like? Does it need the help? Because again, the more we can chop or crush residue, the more cuts, the more entry points for fungi to get in and do the job. There's another thing. So remember, we talked about fungi, we talked about tillage and what that does to soil. So this is a quote that I really like, that when I saw this and learned about it, this surprised me. I thought the susceptibility to soils to compaction was heavy equipment and maybe wet ground. No, it's a biological problem. In particular, low fungi. It's fungi that glue the soil particles together. So let's look at, kind of an analogy. Imagine with me, you've got 2 5-gallon buckets. In one, we're going to take a really finely ground sand. We're going to tamp it down real hard. In the other one, we're going to put golf balls. We're going to try to tamp it down. Good luck. It resists compaction. Now let's pour a gallon of water on top of each. Clearly, it goes right into the golf balls. What happens on the sand? It puddles on top. It's real slow to get in. This is what soil is like. All soil is made up of fine particles called microaggregates. When we have fungi working in conjunction with roots, they produce the glue. They start gluing those microaggregates together into larger particles called macroaggregates. And then the fungal hyphae, that white feathery stuff, and some of the plant roots encase it like the skin on the golf ball. Now we have a stable macroaggregate. They don't pack together as tight. Now there's air and water channels around them. And the glue they make, bacteria love. It is a great food, but when it's encased in a macro aggregate, the bacteria can't get to them anymore. But when we till ground, the first thing we do, we kill fungi. Fungi live a lot longer than bacteria. They're spread throughout the soil, but they don't like to be disturbed. When we till them, we kill them. Plus, when we till, we break those macro aggregates open. Now the bacteria can eat the glue. The golf ball collapses back into sand. So we lose our soil structure, and we can see it. So I live in Fort Wayne, Indiana, and there's a county park not far from my house. It's got trails through it. I like to run through there or jog, as my doctor says, because I'm really slow, and that's okay. It's good exercise. Anyhow, right after Thanksgiving, I had to stop and take a thick picture. This farm, by the way, is organic, and it's a demonstration sustainable farm. Now, there's no cover crop, which is a problem, but look at those clods. They're on the left. They were huge. They were bigger than footballs. I have never seen clods so big. Look at the face. They're sheer. They're solid. This is how we make bricks. And what's happening here is, even though it's organic and they're trying to help the soil, when they tear the ground up like this, there's no fungi. There's no glue. Compare it to the soil on the right. Look at the face where the sun is hitting. All the little bumps, those are the macro aggregates. This came from a customer who's showing off his earthworms, which is great. But if you put that clod in your hand, it would hold water, it would be moist, and you could just crumble it. Not so the stuff on the left. So last summer, we put data around this. We applied product where it had never been applied before in Iowa. We did this across 44 farms, 5,400 acres, and we followed bulk density and water infiltration. So the product was applied when the corn was planted, corn or beans, in April, around the end of April. And we can see bulk density over a 7-week period decrease, and water infiltration versus the check go up 64% in 7 weeks in corn, 53% in beans. I had a guy kind of tongue-in-cheek say, when someone asks me, when I have ground like this, how much rain did you get? I say, all of it. That's what we want, because what's happening is, in this 7-week period, as the roots grow, so are the fungi. The fungi are making glue. We're making the golf balls out of the sand, the macro aggregates out of the micro aggregates. We're making the soil better. Here's another way, and this isn't really looking at soil texture as much, but this is a customer in South Dakota. He started using our product in 2019. Now he's in southeast South Dakota. It tends to be very dry. 2020, and by the way, when you look at the NDVI, green is good, yellow and red is bad in terms of plant health. So purple, by the way, in that lower right corner, that's his house. So this is his home quarter. 2020 was a good year, good moisture. Look at the health of the crop, even in 19. 21 and 22, severe drought. Both years he called me in July, literally, Dave, I'm not going to get a crop. And in October, both years, Dave, I don't know where this crop came from. 2023, he actually won the state Group 2 soybean yield contest, dry land, almost 83 bushels an acre, about double his county average. So fungi do more than this. They protect against what's called abiotic stress. One of those stresses is drought. This is now western Nebraska. You can see where the product was applied and where it wasn't, because fungi are helping the plant not only produce that rhizosheath, but they're signaling the plant how to take care of themselves genetically to mitigate the drought stress. Other kinds of stress we see here, this is the opposite. This is Tennessee. It wasn't hot and dry. It was cold and wet. Look at the difference in emergence and stand. Which one would you rather have? I mean, so much of what your crop is going to do is based on how fast it comes out of the ground and how quickly it gets rooted. So I'm giving you a lot here, so I'm going to kind of try to wrap it up. Another thing the biology does is it protects against diseases. So what happens when you have diverse biology is there's a lot of different food sources in total in most ground, but any particular type or strain of a bacteria or fungus or protist is narrow in what it can actually occupy and eat. But when you have a consortium that protects and feeds each other, they can get into the food sources much more broadly as a community. And so when single strains or other products that have only a few bacteria, which is what most microbial products are, try to come in, they're treated as an invader, just like as a disease would. So we see where we have biologically healthy soils, far less disease. And actually, you could go to the U.S. Department of Agriculture and look up their article on suppressive soils. It's really good. The science is pretty solid. And we see this with customers. So this is actually, just to throw something different out there, blackberries in North Carolina. The ones on the left were not treated. There is disease. There's not a whole lot of immature fruit versus the ones on the right. You see all the red immature berries. They do blacken, obviously, when they ripen, and far less disease, far healthier plants. Another thing biology will do, and by the way, these are pesticide claims, which we're not making. I'm just talking about biology. When we have good, diverse biology, they fight each other. I said there's a farm under your feet. There's also a war. It's a battleground. So they're always fighting for food, for space, for resources. Nematodes, of course, that are plant pathogens, suck the energy out of your plants by feeding on the roots. When we have diverse biology, and this is our product, and we actually have patents on this. This is North Carolina, the Eastern Shore, very sandy ground, some of the highest nematode counts I've seen. 3,000 to 4,000 soybean cysts, over 9,000 root knot. And this was an agronomist that did studies. He got 18 bushels where he didn't have good biology, he did not use our product. Where he used it once, doubled to 30, 35, got up to 45 if you do the second application. Again, microbes can parasitize and kill your plant parasitic nematodes. It's amazing. So if you look at the box, this kind of blew my mind. The microbes in the soil are far more important to protect your crop against insect feeding than the plant's own genetics. So why? Well, first of all, if we think of piercing and sucking insects like aphids, whiteflies, mites, they drink the sap. But what happens is when the plant is healthy, and Brix is a measure of sugar and acids, but sugar in this case, where it's healthier, there's more sugar. They don't like that. And it's because sugar attracts water chemically. And so a healthy plant with a higher Brix or higher sugar sap can kill a piercing or sucking insect because it dehydrates them. So what do? They go to the weak plants. Maybe you've seen this in your field. It's the ones on the end rows, the ones that aren't doing so well. They get all chewed up. They're covered in the insects. Same with the chewing insects. A healthy plant is able to produce more cellulose and lignin. Again, those are the polymers that make your residues so hard to break down. Those are the polymers that make a 100-foot tall tree stand up to a 90-mile-an-hour crosswind. They're also polymers that make the leaf tough to chew. So the insect doesn't like it. They go to the weaker plant. So we get a lot of insect control. The last thing I'm bringing up is yield, and it's on purpose. When you have diverse, healthy biology, your soil has already given you a lot of benefits. Nutrient use efficiency, less disease, better structure. It's more open. It's easier to work. You can plant it faster. You don't have compaction, etc. In that ground should reward you in higher yield. And so if you're curious, we have a lot of yield data on our website. This is across Iowa. This is from Heartland Co-op. My point on fertilizer reduction is important. However, no one can tell you what the ideal NP and K levels are on your ground. Microbes, like our product, are a tool to help you dial in where it pencils out the best. And that's what this third-party trial in Iowa is showing, that, depending upon the cost of fertilizer and the commodity price of corn, you can see even 20% less N, P, and K, we still had a little bit better yield, but where does it pencil out? Remember, there's about half a pound of nitrogen in a bushel of corn. If you're putting on a pound or more, you're double or more the removal rate. You can start backing down. How much? I would do 10 to 20% bites, N, P, and K. If you got light sandy ground, you may get maybe down to 0.8 tenths of a pound of nitrogen and not impact your yield. If you've got heavier ground, you could get closer to half a pound. The guy in South Dakota that I showed earlier, he's at 0.55 pounds of nitrogen, no P or K, and his yields are still 220 bushel an acre corn, but he did that over 4 years. He has heavy ground that has had cattle manure spread for years, so he can get away with that. He can tap into that rich soil bank. What you can do is really, this is a tool to help you figure that out with your agronomist. Again, yields in cotton. So look, you earned your credits. This is a lot. So, and it's a lot to absorb. There's a couple things to remember to make it simple. You want the microbes, the diversity in your soil. Bacteria, actinomycetes, fungi, protozoa, amoeba. How can you tell if you're getting a product like that? Well, if it's a dry powder, it can't have all this biology. It may have some fungal spores, but it will primarily be bacteria spores, which is only a slice of all the bacteria. If it's a liquid in a tightly sealed container, stored at room temperature, they're microbes that do not need oxygen. This means you might have, again, a narrow slice of bacteria, maybe yeast, but you're not getting the white fuzzy fungi, you're not getting any protozoa or amoeba. That will not replenish your soil engine. We make a product that's refrigerated. It's the only way we can stabilize it. But when you go to the trouble of having diverse biology, remember, you're developing soils that help you grow a better crop with fewer inputs while you actually increase the value of your land because you're making your soil better. Which ground would you rather buy? So I'll stop here. I like these shirts. This is a customer. He made them, and he kind of made the comment, wow, I'm improving my soil. So it's Holganix, so the farm is here for me, for his kids. And by the way, we're expanding. So we have distribution, but we're looking to add more, and we're hiring agronomists. So there I put in my little plug. So Robin, with that, I will stop and get back to the title of Ask the Scientist, because I talked a long time, so I'm going to be quiet until we get questions.
Well, thank you, Dave. I feel like I just sat through my soil science class, the entire quarter's worth. But it was really educational. So we do have questions, though, so I'm going to start with the first one here is, how long do the microbes in your product persist in the soil?
That's a fantastic question. And we're working with a company that actually is doing those studies. So first of all, I expect that they will change through the growing season because that's normal. We have some microbes that are called lactobacilli that are actually found in yogurt. They're in our gut. I don't think they persist very long, but they give a nice burst of organic acids that releases some of the locked up nutrients like P and K. Our fungi, from what we've seen last year, are surviving and growing throughout the entire growing season. So we improve the bacteria to fungal ratio. How much they overwinter depends on how much you disturb the ground and on what happens with air and moisture. Moisture and temperature. So year after year, we don't know. We do know that you can see improvements in the soil from year 1 to year 2 to year 3. So the changes, it's a marathon, not a sprint, but we see benefits every year. And by the way, depending where you are, we come out to the field, we measure the soil, we develop an online site for you to access your data, and we don't charge you for that. So most of the places in the Midwest, we do that. Now, just to stand by the product to give you the confidence that adding life back to your soil will help you produce more, use less, and make a better profit.
All right. Our next question is, I Y drop my 32% at the base of the corn row. Does that concentration of nitrogen set back my biology that's helping the corn?
Probably not. It depends upon the moisture levels that you've got in the ground, because that is a relatively high salt fertilizer. But the microbes, by the time you're Y dropping, they colonize all along the roots. So you may disturb some at the very, very surface, but I wouldn't be too concerned about that.
All right. Our next question, actually, it seems like a pretty good one because I'm not sure you mentioned it. How does soil pH affect the activity of the Holganix products?
Well, that's a great question. So microbes in general like the same pH range as your crop, but they have a much wider tolerance. So the biology through time will bring the pH back to a more neutral range. And, the 6 to 7.5, it is not a quick fix. But if you have a very low pH, we have especially bacteria that secrete different things to chelate the micronutrients, because things get tied up at the extremes. So they can still tap into it. They're not going to work as well at the low and the high, but they still do work. And I've got people with pH extremes that use our product, and they see really good results. My advice, though, scientifically is if you're very acidic or very basic, do something to fix that, because there's a lot more going on.
Okay. And this next person has asked two questions. The first one is, what is the ideal fungi-bacteria ratio, and what is the ideal protist-bacteria ratio? And I'm sure it depends.
Those are phenomenal questions. And I've seen people say, and people even in our company will say, the ideal bacterial fungal ratio is 50-50. I don't see the supporting science. Remember, the fungi are much heavier and much larger than bacteria. So are we measuring by weight? If it's by weight, the fungi should actually dominate over bacteria. Protists should always be a much lower amount, but I don't have a good ratio. But knowing that a protist can eat 10,000 bacteria a day, we want to see lower protist numbers, just not zero. So you're asking the right question. I just haven't seen the science that tells the right answer, other than they're all important.
Okay. All right. Let's see. Our next question here is, how long does the product need to be refrigerated, and is it delivered to the field cold if it does need to be refrigerated?
So after we make it, we quickly chill it to 37 degrees, and we hold it there. That keeps the microbes, basically puts them to sleep. So at 37 degrees, it's good for 2 years. And yes, we ship it to the farm cold. And when it comes in totes, we have an insulated wrap. And depending on the outside temperature, store it out in direct sunlight, out in the barn or something, you get anywhere from 4 to 8 weeks easily. So here's the thing. Here's another clue in terms of what's in your microbial product. Our product has a pound of molasses in every gallon. And the founder said, and he's right, it smells like a mix of Sam Adams, glazed donuts, a hint of vinegar. And it does. It actually smells pretty good. If it's out too long, you will not get your nose next to it. The oxygen is used. It's like your ground that's been water saturated for more than several days. It's like the rotting swamp water. For our company, so that means a lot has died, and now we only have those bacteria that can live without oxygen and some yeast strains. It smells so bad. You can use it, but that's not what you paid for. It doesn't have the diversity. We replace that at our expense, yes, including shipping and handling, so no extra charge, no questions asked. Sometimes it happens. It's rare. But I had a guy earlier today say, hey, I had a jug down in my basement the last 2 years. That was his fault. We're sending him a fresh one. I don't care. We stand by our product. Don't worry about refrigeration.
All right. Let's see. What soil temperature is needed to take the pre-application microbiometer test? Is there an optimal temperature for that?
That's also a good question. I'm not sure it makes a whole lot of sense below 50 degrees Fahrenheit because things aren't really growing. I guess you would get a baseline, but to me it would be more informative. If the soil's warm enough to germinate a seed, the microbes are starting to grow too. So to me, that would be a better snapshot. But once it's cold or frozen, I'm not sure it makes a whole lot of sense.
Okay. Another listener would like to know how does the Holganix product work with cover crops?
Oh, I'm glad you brought that up because I said microbes help, enable cover cropping. Remember that picture I showed of the cold, wet soil from Tennessee? That's what it's going to help because so many places the race is with a cover crop between harvest and winter. So diverse biology, like in our Holganix Bio 800 Plus, helps with germination and root development. So we get that cover crop established faster. And that can make the difference between having a good stand and a really poor one before winter comes.
Okay. And here's someone just asking for a clarification of that you said some beneficial fungi use lignin as a food source. And I believe you did say that. Yeah.
Yep. They are.
All right.
They tend to be white rock. The pseudomycetes, if you really care. They tend to be what? Dirty.
Let's see. The questions are coming in thick and fast. Do the calcium, magnesium soil levels impact the soil microbiology?
If they're really low, because remember, microbes need those minerals as much as your crop. The difference is the microbes can tap into that total pool that your plant can't. I've seen a lot of ground, though, actually in the last year, more high magnesium ground, and I have no explanation why. The microbes, because we go out with a Crystal Labs probe that looks at the nutrient profile, and it uploads to the phone in 30 seconds. It's amazing. It does bulk, it does bulk density, pH, moisture, and all the different nutrients in a microbiometer test. The microbes are still doing well in a high mag ground. I just, again, if it's really, really low, my bet is unless you don't have any in the total pool, then the microbes are tapping in, and they'll eventually feed it back to your crop.
Okay. Let's see. Someone's asking, how does Holganix compare to regionally grown compost? Oh,
regionally grown compost.
So, they didn't say manure, they said compost.
Yeah, so compost, by the way, is a great food source for Holganix because our product is based on a compost extract. We just have a very defined manufacturing process and starting material that's only plant-based. So we're only getting microbes that are associated with plants. There's no manure, so animal waste, so we avoid the pathogens in that, the human pathogens in particular. Compost is highly variable based on what you put in it and how well it's sterilized in the process. So compost is fantastic. Like I said, it's a great food source for Holganix, which really puts all the biology that might be missing in any given compost back into it. Personally, around my house, I have a guy I work with. He's about 15 minutes away. He collects compost from local farmers. It's the bedding from cows and horses. He collects food waste from the city of Fort Wayne and then throws yard waste on it. And it's a beautiful compost. And when I put Holganix on it around my yard and his customers, it's amazing. The combination is, again, blows me away.
So it's not an either or, it's an in addition to.
It's a both. Yeah, and by the way, we
Okay.
make our product. We have 11 different climate zones, soil types across the country, so we do use native microbes in our product. Although
Okay.
I have to
That's
say,
good.
I really pay attention to this scientifically. It feels like you want native microbes. Scientists are still scratching their head as to how important that is. We know we don't want to introduce a pathogen that's soil-borne that isn't already endemic. That's easy.
Mm-hmm.
But everything else, the jury's still out. So maybe next year when I do a webinar, I'll tell you more about that if there's more that we've learned.
All right. Well, I think we are out of time. We have a few more questions that didn't get answered, but we do have your email and contact information, so Dave can reach out and answer specific questions that you had that weren't answered. So we're sorry we didn't get to that. But I'm going to wrap it up now and thank Holganix and, of course, David Stark for doing this excellent webinar. As a reminder, an on-demand version it will be archived in the next few hours, and you can access it at the same login that you used to access it today. And you can also, you'll get a reminder tomorrow around the same time that will also have the link in case there's something you want to go back and see. And you can also go to croplife.com/webinars and find it there. So lots of chances to go back and get things clarified. So again, I want to thank everyone for your time today and obviously to Holganix for making the webinar possible. Thank you so much.
Yeah, thanks everyone.